Okay, let's unpack this. Welcome back to the deep dive. Today, we are taking a massive intellectual leap back in time. I mean, way back. Before life, before cells, before even the Earth as we really know it. We're trying to figure out how the most fundamental molecules, the ones that define us, actually came into existence from, well, plain non-living matter. We are diving deep into the chemistry before the biology. And our sole source for this journey is Chapter 6 of Strickberger's Evolution. It gives us the essential roadmap for tracing that earliest, abiotic phase. Our mission today is to trace how simple, non-living chemical interactions sort of laid the groundwork for complex biological structures and ultimately, well, set the stage for natural selection.
It's a staggering story, really. And to understand it, we need to know the central players first. We are looking at two fundamental molecular classes, absolutely necessary for life's continuity. You can think of them as like the information library and the machine shop. So first, the nucleic acids. This is your library. You've got DNA, which stores the permanent blueprints, right? And then RNA, which are more like the temporary copies, the messengers, the regulators use to actually execute those instructions.
Okay, the library. And the machine shop.
That would be the amino acids. These are the real workhorses. They're the building blocks that link up to form peptides, and then they fold into proteins. And proteins act as the enzymes and catalysts that, actually make things happen inside a cell. They're the tools, the assembly line, the workers, all of it.
Got it. Library and machine shop.
And the beauty is the necessary ingredients. They weren't particularly rare. Our analysis shows that the starting line was marked by this high cosmic abundance of basic elements: hydrogen, oxygen, carbon, nitrogen, sulfur, phosphorus, calcium, the essentials.
Right, and on the early, chaotic Earth, we had gases venting from the interior, things like hydrogen gas, methane, constantly being bombarded by just massive amounts of solar and UV radiation. That energy hitting those simple gases, it sparked the formation of early organic molecules, things like ammonia and hydrogen cyanide.
Exactly. It was, surprisingly perhaps, a kind of universal organic chemistry already in the making.
Universal? How so?
Well, that universality is maybe the most important insight here. The chemical rules themselves were sort of favoring certain outcomes globally, not just on Earth. This led to a pre-biological period we now call molecular selection, where stable, efficient molecules were just chemically favored over unstable ones.
So like a chemical survival of the fittest before life even started.
Precisely. This was the very first filter, the true precursor to natural selection, operating long, long before the first living cell ever appeared.
Okay, so if the early Earth was so chaotic, how can we possibly know what ingredients were actually around? Seems like guesswork.
Well, here's where it gets really fascinating. We don't entirely have to guess, because space, believe it or not, delivered us a sample.
Delivered a sample? What do you mean?
Meteorites. Specifically, carbonaceous chondrites. These are meteorites that contain carbon compounds.
Right, I've heard of these, like the Murchison meteorite, the one that fell in Australia back in '69.
That's the most famous example, yes. And the research on it was just astonishing. About 1% of that meteorite turned out to be organic carbon. Inside, they found over 14,000 different chemical compounds, including crucial building blocks like purines, pyrimidines, glycerol, and dozens of different amino acids, glycine, alanine, many others.
14,000, wow.
Yeah, and the insight here is massive, these extraterrestrial amino acids. They were structurally identical to those generated in lab experiments simulating early Earth conditions.
So wait, that means?
It proves that this kind of organic chemistry isn't specific to Earth. It's a common outcome across the cosmos, likely preceding the formation of our own sun and planet. Oh, and get this. The Murchison meteorite also carried fatty acids. And when you put those in water, they spontaneously form these little membrane-like sacs, rudimentary bounded vesicles.
Okay, hold on. So we have the building blocks, the amino acids falling from space, basically. And we have early membranes, these fatty acid sacs forming spontaneously. If we have the necessary molecules and some kind of physical boundary, what is the hard line between non-life and life then? It seems blurry.
That really is the big intellectual hurdle, isn't it?
Yeah.
And the source material reminds us of Erwin Schrödinger's influence here, back in 1944. He framed the whole problem in physical terms, which really kick-started the molecular biology revolution. Our analysis suggests life requires, well, 5 key attributes, but the bare minimum for life's continuity. That requires an integrated system. You've got to be able to survive and replicate, obviously. Perform metabolism, transform external energy for growth and maintenance, exist as individuals or populations.
Right.
Based on some kind of membranous structure, like a cell. And crucially, use that complex integrated system of DNA, RNA, and proteins to run the whole show.
So all four molecular components plus the cell.
Exactly. Without all of those working together, continuity fails. You don't get life carrying on.
Okay, let's look closer at the structures then. Starting with those workhorses, the amino acids. Can you sort of paint a picture of how they're structured?
Absolutely. Every amino acid has a common backbone. There's a central carbon atom, we call it the alpha carbon. It's connected to three things that are always the same: an amino group, a carboxylic acid group, and just a simple hydrogen atom.
Okay, invariant features.
Right. But the fourth connection is the R group, the variable side chain. This R group is the key bit. It determines the specific identity and the chemical behavior of that particular amino acid.
And when you string these amino acids together using peptide linkages, you get these massive polypeptide chains that then fold up into functional proteins. That's the machinery.
Precisely.
Okay, now for the library, the nucleic acids. The building blocks here are different. They're the nucleotides. Each one has a 5-carbon sugar, a phosphate group, and then one of 4 nitrogenous bases: adenine, guanine, cytosine, and thymine, or uracil in RNA.
Right. A, G, C, T, or U.
What's just staggering is the information capacity. With 4 possible bases, if you have a chain of, say, n nucleotides long, you have 4 to the power of n possible different messages you can encode.
It's an almost unimaginable amount of information storage potential.
Truly astronomical.
It is. And unlocking how that potential was actually realized and used, that was the great moment of 20th century biology. The ultimate breakthrough, of course, came in 1953 with Watson and Crick, aided immensely, we should always add, by Rosalind Franklin's absolutely critical X-ray crystallography work.
Yes, absolutely.
They revealed the double helix.
That iconic structure. But what does it fundamentally tell us about heredity, about passing information on?
It tells us two key things structurally. First, the two strands run in opposite directions. They're anti-parallel, and they're wound together like a spiral staircase, a right-hand helix. Second, and this is the crucial part, the strands are held together by complementary pairing. This was dictated by Erwin Chargaff's earlier vital finding Chargaff's rule.
Chargaff's rule, that's the ATGC thing?
Exactly. The amount of adenine always equals the amount of thymine, and guanine always equals cytosine. So A always pairs up with T across the helix, and G always pairs with C.
Ah, so that chemical matching, that parity, that's the structural key.
That's it. And that structure immediately suggests the mechanism of heredity. Because A always pairs with T and G always with C, each single strand serves as a perfect template for building a new complementary strand.
Like a mold.
A perfect molecular mold. You unzip the helix, and each separated half guides the construction of an identical partner strand based on those pairing rules. The result is exact molecular replication.
So the genetic message can be transmitted faithfully, generation after generation. Wow.
That's the foundation of heredity, right there in the structure.
Okay, so we have the stable information stored in DNA and the ability to copy it precisely. The next logical step is getting that information out to build the protein machinery we talked about earlier.
Yes, we call that the information flow, or sometimes the central dogma of molecular biology.
Right.
This is where the process becomes more like a multi-step factory line. DNA is replicated to make new DNA, obviously. That's step 1 for cell division. Then, for making proteins, you have transcription. This is where a specific segment of the DNA, a gene, is copied onto a sort of disposable messenger molecule, messenger RNA or mRNA.
Okay, so like pulling a specific blueprint from the main library and making a temporary construction note.
That's a great analogy. And that construction note, the mRNA, is then shipped out to the ribosome for step 3, translation. This is where the messenger RNA sequence is read, and it's converted into a specific chain of amino acids, which then folds up into a functional protein.
This is where the machine actually gets built following the instructions.
Exactly. And the language used in that construction note, that mRNA sequence, is the universal genetic code. Pretty much, yes. It was largely deduced by Crick and his colleagues back in 1961. They figured out that the code has 4 really key features. First, it uses non-overlapping triplets of nucleotides. 3 bases read together. These are called codons. Each codon specifies an action, usually an amino acid or a start-stop signal.
Okay, triplets, not single bases, not pairs, but groups of 3.
Correct. Second feature. Reading the message requires a specific start codon, which is AUG, to signal begin translation here. But there are 3 specific stop codons, UAA, UAG, UGA, to signal end of protein.
Start and stop signs. Makes sense.
Third, and this is really critical, the code is degenerate, or you could say redundant.
Degenerate, redundant, that sounds bad, like it's inefficient or prone to errors?
You'd think so, but it's actually the opposite. It's an incredible defense mechanism. This redundancy means that most amino acids are coded for by more than one codon.
Okay. How does that work?
Well, our source material emphasizes that if you look closely at the codon table, you'll see this redundancy is mostly concentrated in the third codon position. For some amino acids, the first two bases of the triplet are fixed, and the third base can be almost anything, any of the four bases sometimes, and you still get the same amino acid coded.
Ah, I see. So a mutation in that third spot might not even change the protein.
Often it doesn't, which brings us to one of the, I think, greatest insights in molecular biology.
Which is?
Francis Crick's wobble hypothesis, proposed in 1966.
Wobble.
Wobble. The wobble hypothesis explains how this redundancy works efficiently. It states that there's relative non-specificity or wobble pairing allowed at that third position when the mRNA codon interacts with the transfer RNA, the tRNA, during translation. For example, a single tRNA molecule, the one carrying the amino acid, doesn't always have to perfectly match the third base of the mRNA codon. There's some flexibility, some wobble.
Give me an example.
Okay, the base inosine, which is often found in the tRNA anticodon loop, it can actually pair up with U, A, or C in the third position of the mRNA codon. Guanine in the tRNA can pair with U or C in the mRNA.
Wow. So one tRNA can recognize multiple codons for the same amino acid.
Exactly. This is just a brilliant evolutionary economizer. It does two things. It minimizes the impact of translational errors. A mutation in that third wobble base often results in the same amino acid being incorporated anyway. And it reduces the total number of different tRNA molecules the cell needs to make, making the whole translation machinery more compact and efficient.
That's incredibly clever. It's amazing to think that this complex, error-minimizing code is almost entirely universal across all known life. You find the same code in bacteria, in plants, in us, with only very rare exceptions, usually in mitochondria or some basal eukaryotes.
That universality is profound. It leads to concepts like frozen accidents.
Frozen accidents, meaning what?
Meaning, once a system like this, with maybe 20 specific amino acids incorporated into this precise coding and translation machinery, becomes established and functional, well, changing it becomes almost impossible.
Because any major change would mess everything up.
Exactly. It would cause widespread protein malfunction and likely be lethal. So the code essentially got frozen in place very early in life's history. Further evolution then focused on optimizing around that code, selecting for mechanisms that minimize error, favoring certain codon frequencies, making translation super efficient, but not changing the code itself.
Right. Okay, so we have the molecules, the code, the replication. Now we shift focus a bit from how these molecules work to where on the early Earth they might have actually been first manufactured abiotically.
And the source material suggests a good way to infer possible locations is by looking at modern life that thrives in extreme conditions, places we once thought were uninhabitable.
You mean like extremophiles?
Exactly. Particularly thermophiles. These are heat-loving organisms. Some bacteria and archaea can survive, even thrive, at temperatures up to 320 degrees Celsius near deep-sea vents or in volcanic hot springs. The very fact that life can survive these incredibly stringent conditions today suggests that maybe, just maybe, life originated in similar environments long ago.
Okay, that makes sense. So what are the prime candidates for these early Earth factories? Site number one mentioned is volcanoes. Early Earth was volcanically hyperactive, way more than today, and this constant activity actually drove mineral evolution. Apparently there are like 4,700 species of minerals now evolving in stages linked to geological changes.
That's right.
And the high greenhouse temperatures back then from CO2 and methane actually helped molecule formation. Plus, volcanic sediments produced these sponge-like minerals called zeolites.
Zeolites, yes, they have intricate porous structures.
Right, and they could apparently trap, retain, and maybe even catalyze reactions between organic compounds, like little mineral reaction chambers.
That's one strong possibility. Then site number two, deep sea vents. These were only discovered in 1977, which is pretty recent, geologically speaking. These hydrothermal vents spew out superheated water, sometimes up to 350 degrees Celsius, rich in minerals and chemicals from the Earth's interior. The hypothesis is that these could have been sites for synthesizing amino acids and ammonia.
What makes them so suitable?
Well, the environment highlights the potential role of early, simpler catalysts. Before complex enzymes evolved, maybe simple metallic ions like iron 2+++++ played a key role.
Like primitive enzymes.
Sort of, yeah. And the source gives a great comparison. Look at the efficiency of catalysis. Phe++1 at IR alone have some catalytic activity. incorporate that iron into a heme group, like in hemoglobin, and efficiency jumps. Put that heme into the full enzyme catalyze, and the efficiency increases by 1,000,000,000-fold.
Wow, 1,000,000,000-fold.
It shows a plausible evolutionary pathway from simple inorganic catalysts to the hyper-efficient enzymes life uses today. And this idea gained support from experiments by Huber and Watershauser in the late 1990s. They managed to generate amino acids, like alanine, from simple precursors like pyruvic acid and ammonia under high-temperature, high-pressure conditions, mimicking those vent environments.
Okay, volcanoes, deep-sea vents.
Yeah.
What's site number 3?
Site number 3, clays. This is a really elegant theory, I think.
Clays, like mud.
Well, specific types of layered clays, particularly one called montmorillonite. The hypothesis is that the layered structure of these clays could have acted as the first kind of self-replicating surface or template.
A mineral scaffold for life.
Something like that. And the evidence is actually quite compelling. Lab studies show that clays can catalyze the condensation of nucleotides into short chains, maybe up to 55 nucleotides long. They can also help polymerize amino acids into simple peptides. So this clay surface could have provided the necessary structure, the solid-state platform, that might have been essential for organizing the complex steps needed for the early evolution of a protein synthesizing system. Concentrating molecules, facilitating reactions.
Bringing things together that were just floating around.
Exactly. Overcoming the dilution problem of the early oceans.
Okay, those are the locations. But we can't talk about the chemical plausibility of abiotic synthesis without mentioning the two absolute giants in experimental confirmation, Miller and Fox. We really need to give these experiments their historical weight.
Oh, absolutely. They are cornerstones. First, you have Stanley Miller's 1953 experiment. Hugely famous. He simulated what was then thought to be the early Earth's atmosphere in a closed flask, basically methane, ammonia, hydrogen gas, over boiling water to simulate oceans.
And then he zapped it.
Yep. Zapped it with an electrical discharge, mimicking lightning or intense UV radiation.
And the result was, well, it was a worldwide sensation, wasn't it?
It absolutely was. Proof of concept. He generated a whole slew of organic molecules, including over 20 different amino acids, urea, other essential compounds found in life. It showed, pretty convincingly, that just putting simple energy into a simple, plausible early atmosphere inevitably results in the spontaneous formation of life's organic building blocks. No magic needed, just chemistry.
Incredible. And then Sidney Fox in the 1950s, he took it kind of the next step, right? Polymerization.
That's right.
Yeah.
Fox focused on how these building blocks might link up. He demonstrated that if you simply take dry mixtures of amino acids and heat them moderately, say on a simulated primordial volcanic slope, you can generate what he called proteinoids, or thermal proteins. These were polymers, chains of amino acids, that actually showed some protein-like features, like forming little spheres or having weak catalytic activity. It proved that polymerization, linking these monomers together, doesn't necessarily require complex cellular machinery either. Simple environmental conditions like heating and concentration could be sufficient for that crucial step.
Okay. So wrapping this all up, what does this journey through Chapter 6 really tell us? What's the big picture? It seems our journey shows that the continuity of life, everything that lives today, relies on this incredibly sophisticated, integrated molecular machinery of DNA, RNA, and proteins working within cells. That's non-negotiable. But the molecules themselves, the amino acids, the purines, the pyrimidines, the basic units, they trace their origins way, way back to high cosmic abundance, back to chemistry happening in the early solar system, maybe even before Earth, and certainly to purely abiotic interactions on a dynamic, high-energy early Earth.
That's it exactly. The chapter beautifully stresses that our understanding of the origin of life's molecules is fundamentally rooted in physics and chemistry. We're essentially connecting the dots. From completely non-living materials, whether they arrive here on a meteorite or bubble up from a deep-sea vent, or form in a primordial soup zapped by lightning. Connecting those simple beginnings to the incredibly specific, redundant, error-correcting, and nearly universal genetic code that defines every living thing we know.
It's quite a connection, which leaves us, as always, with a final provocative thought for you, our listeners. This one builds on a theory mentioned regarding collective non-Darwinian evolution proposed by Vitzigian and colleagues. We noted the genetic code is almost perfectly universal. We talked about frozen accidents. But how did it become universal? Was it simply fixed very early in one lineage and then passed down vertically parent to child out competing everything else?
Or was there another way?
Right. Consider this hypothesis. What if the code's universality wasn't initially due to strict vertical inheritance, but rather was stabilized during a long, early period of extensive horizontal gene exchange?
Imagine early proto-organisms, maybe not even true cells yet, freely swapping genetic material, sharing innovations.
Yeah, sharing the best code, the most efficient translation tricks across different emerging communities, kind of like collaborating on the operating system before distinct species or lineages really lock things down with hereditary barriers.
So the code became universal through widespread sharing and consolidation before vertical inheritance became the dominant mode.
Something fascinating to think about anyway, as you contemplate the fixed ancient language that's currently directing the machinery inside every one of your own cells.